Now that we know that thin filaments slide over thick filaments during muscle contraction, let's examine how the process works in detail. Myosin molecules have two important features that enable the thick filament to bind to and move the thin filament. First, the hinged portion of the tail allows the myosin head to reach the binding sites on actin. Second, the myosin head can pivot back and forth between high-energy and low-energy positions. This pivoting motion constitutes the power stroke that pulls the thin filament along the thick filament. A sarcomere shortens when myosin heads on the thick filaments form cross bridges and pull on the actin molecules in the thin filaments. The process known as the cross-bridge cycle consists of 4 steps. 1. Cross-bridge formation. 2. The power stroke by the myosin head. 3. Cross-bridge detachment. And 4. Reactivation of the myosin head. Let's examine each step. Step 1. Cross-bridge formation. The cycle starts when excitation-contraction coupling causes the myosin binding sites on actin to be exposed. Myosin heads bind to these sites, forming cross bridges. Myosin starts in its high-energy state, in which the ATP binding site on the myosin head is occupied by ADP and inorganic phosphate. The myosin head is ready to pivot for the power stroke. Step 2 is the power stroke. Release of inorganic phosphate from the myosin head causes the head to pivot, pulling the thin filament toward the center of the sarcomere. ADP is then released as well. The myosin head is now in its low-energy state, but it remains attached to actin. The energy for the power stroke is ultimately provided by the ATP that binds to the myosin head. Step 3 is cross-bridge detachment. Myosin remains attached to actin until an ATP molecule binds to the empty ATP binding site on the myosin head. This weakens the link between the myosin head and actin, and the myosin head detaches. The detached myosin head remains in its low-energy state. Step 4 is the reactivation of the myosin head. This occurs when the ATP bound to the myosin head undergoes hydrolysis to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, pivoting it back to its high-energy state. The cross-bridge cycle starts again when the energized myosin head forms another cross-bridge. As long as the myosin binding sites on actin remain exposed and ATP is available, the cross-bridge cycle repeats, pulling the thin filaments toward the center of the sarcomere. During muscle contraction, cross-bridge cycles occur between the thick and thin filaments along the entire length of each sarcomere. But not all myosin heads are bound or disconnected at the same time. They can be in different steps of the cross-bridge cycle. Remember that there are thousands of sarcomeres in a myofibril and thousands of myofibrils in each muscle fiber. Cross-bridge cycling at all of these sarcomeres causes the entire muscle to contract.